Hi there. This video is called Molecules of Life, and I'll be focusing more on nucleic acids such as RNA and DNA, and just glance over proteins and some other molecules of life. I do cover these in a cell biology course, so there are some videos that go into more detail about some of those molecules as well. So in the previous lectures and videos, I talked primarily about the evolution of Earth, including physical characteristics and chemical characteristics, and a bit on some of the major evolutionary events on Earth. Now I want to get a little smaller and talk about specifically some of these molecules, including DNA and RNA. And I'll start with DNA because most people are more familiar with DNA. Before I talk about some of those molecules, I just want to introduce some perspective on all of this so you realize how small these things are and how rapidly things are going on, and how many of these approximately are in a cell. So if you take a cell and expand it by about 1,000,000 times, so a single cell 1,000,000 times, it's about the size of a classroom, an auditorium, your room, depending on the cell. And so that's what's illustrated here. So on that level, then people would be about this size in that room compared to some of these molecules and organelles. So if we just look at the cell wall and the cell membrane, and this would be for a plant cell with a cell wall, then the wall would be approximately the thickness of a wall, a block wall or a wood wall. The cell membrane would be about 1/2 centimeter thick in that scale, and then there would be proteins embedded in that membrane, and they would coat all surfaces, all internal surfaces. So if you're in this plant cell, then it would be on the floor, on the walls, on the ceiling, everywhere. And then if you go inside the nucleus would be approximately 12 feet, somewhere around that, across. And it would have a double membrane, but the membrane is folded around on itself rather than being a true double membrane. And then nuclear pores would be embedded in that, all around through that large nucleus. The nucleolus would be inside of that, about approximately in diameter, about 4 or 5 feet across. Then you go to the chloroplast would be about twice the height of a human in this scale, about 12 feet by about 4 or 5 feet across, and there would be several of them in this cell. Mitochondria would be a little smaller than that, about the size of a bacterium or a mitochondrion, and on this scale about the size of a human, rather robust human in that case. And then in the nucleus, you would have DNA, and DNA would be about the thickness of some twine, so fairly thick. And if we stretch this DNA out, in humans, you have 46 chromosomes, or 23 pairs of chromosomes. If you stretched out each chromosome, then it would reach from approximately Bowling Green to Toledo, which is about 20 miles. So wherever you are in the U.S., it would be about 20 miles per chromosome, and there are 46 of those. All in all, if you put them end to end, the entire human genome would reach from Bowling Green, Ohio, or Toledo, Ohio, to New York City and back. So a lot of DNA that is gathered within that nucleus. Along the DNA, you would have different proteins, including DNA polymerase, shown here, and then from that, the RNA would be coming off of that, being produced by transcription. That would be single-stranded, so it's about the thickness of string, so about half the thickness of DNA, and it would be folded up, ready for translation on the ribosomes. And speaking of ribosomes, out here in the cytosol, they would be about the size of golf balls on this scale. And in a typical bacterial cell, there would be approximately 10 to 60,000 of them. And in a eukaryotic cell, there would be anywhere from about 1 million to sometimes 10 million, depending on the size of the cell and the amount of translation that it is doing at the time. Then a large protein would be about the size of a lima bean, and there would be, millions of those. And small proteins the size of a pea. An atom at this level would be about the size of a dust speck. So you could see things like sodium atoms and sodium ions and things like that. So now imagine all of those things floating around the room around you. The millions of golf balls, the millions of other proteins, a large nucleus, several chloroplasts, and several mitochondria. Maybe some viruses have invaded the cell, maybe some bacteria have invaded the cell, and now shrink that down to its actual volume. So in the case of our building here, a fairly large building, the Biological Sciences building, you could shrink that down, and if you put about 25 cells together, you could shrink that down until it looked like a small dust speck on your desk or on your table. In the case of if you're in a house, you could probably shrink it down to a dust speck by shrinking it 1 million times. And you'd still just be barely able to see it on your desk or on your table or wherever you are right now, in front of you on the computer, in fact. So you could actually visibly see it at that size. But again, that's only if you put 25 or so of these cells together. You would be able to see those as a dust speck. So that sort of gives you some perspective on the sizes of these things and how many of them are within a cell. So now I want to start talking about nucleic acids. So the basic structure of a nucleotide is you have a pentose ring, either a ribose or a deoxyribose ring, and then a base, either A, G, C, T, or U, and a phosphate group on the 5' carbon of the pentose. So in a little more detail here, you can see the pentose, either a ribose or a deoxyribose with the 1' carbon here, 2' carbon here, 3' carbon here, 4' carbon there, and 5' carbon there, and this ring is joined together by an oxygen group here. In the case of RNA, this hydroxyl would be on the 2' carbon, and in the case of DNA, there would be a hydrogen instead of a hydroxyl group on the 2' carbon. So DNA is actually called 2' deoxyribose. And then on the 5' carbon, hooked together by a diester bond, is a phosphate group. So here are those two sugars. Here's ribose, and again 1', 2', 3', 4', 5' carbons, and a hydroxyl on the 5', 3', and 2' carbons. Whereas 2' deoxyribose has hydroxyl groups on the 5' and the 3' carbons, but only a hydrogen. And this leads to the capability of DNA can fold up tighter than RNA because it lacks that bulky oxygen. Here are the bases in DNA and RNA, the two purines up here, adenine and guanine, and you can see how the rings are labeled. Here's the 1, 2, 3, 4, 5, 6 positions, 7, 8, 9 over here, and similarly for guanine. The pyrimidines have only a single ring, so there's 1, 2, 3, 4, 5, 6 here, and similarly for thymine. For uracil in RNA, which substitutes for thymine, there's a hydrogen here, and the methyl group is not present in RNA. Here's a different view of those. Here's adenine and guanine, and I put a star here on the 9 nitrogens in this place, the 9 position, which is where these two molecules are attached to the ribose or deoxyribose on the number 1 carbon. And here's cytosine and thymine, and again, the star here indicates where those are attached to the sugar group, or the ribose, in that case, to the number 1 carbon in both cases. And again, for uracil, which is an RNA, this methyl group would be missing. There would be just a hydrogen on carbon 5. So a bit about the nomenclature, because I've been using different words here. The nucleobases, just the purines and pyrimidines, and again, you can find some of these in meteorites. It's adenine, guanine, cytosine, thymine, and uracil. Once they're attached to the sugars, then the terminology is slightly different. Adenosine, guanosine, cytidine, thymidine, and uridine. But in most cases, I'll just say A, G, C, T, or U. So here's a bit more about the structure and how they fit together. So here are the pentoses, ribose with its hydroxyl here, and right here on the 3' and 5' parts of this molecule, which is where you have the backbone eventually of the DNA or the RNA. And then on the 1' carbon is where they're attached to the base. And in deoxyribose, 2' deoxyribose, there's a hydrogen here rather than a hydroxyl. For the purines, there are a few differences. Adenine has a hydrogen here, where you have an amino group in guanosine or guanine. And up here you have an amino group in adenine, whereas you have a carbonyl oxygen on guanine. The pyrimidines are here, C, T, and U, and again, there's simply substitution of an amino group up here for a carbonyl oxygen here. And then the difference between thymine and uracil is a methyl group on thymine, which is not found on uracil on the number 5 carbon. And the stars down here indicate where those are attached to the pentose sugar, and each one of these little asterisks on here also shows where they will pair by hydrogen bonding in a typical Watson and Crick pair. So an A will bond to a T or a U, and a G will bond hydrogen bond to a C. But as I'll show you in a few minutes, there are other possibilities. There are other bases in DNA, including a methyl A and a methyl C. In the case of methyl A, there's a methyl group on that part of that amino group up there. And then in methyl C, it's on the 5' carbon. You'll have a methyl group up there. Although adenine appears in both DNA and RNA, its functions in RNA are very much different than in DNA. So up on top you have adenosine triphosphate, known as ATP, and this is a RNA compound that sends energetic phosphate bonds throughout the cell for various purposes. Whereas here's a DAMP, deoxy AMP in a nucleotide, which would be in DNA, and it's pretty much, unreactive. It doesn't perform the same function as ATP in RNA. The nucleotides in DNA and RNA are joined up in the same way. That is, the 5' carbon holds the phosphate group, and then you go through the 4', and then the 3' carbon is attached to another phosphate group, and then there's the 5' carbon on the other side, and going through the 4' and again the 3', eventually ending in a hydroxyl. And in the case of DNA, these two on the 2' carbons would both be hydrogens. In the case of RNA, one of those would be a hydroxyl. And these are joined up in DNA because you can pack those better in the 5' to 3' direction on one strand. Those will pair with the complementary bases on the other side, in this case T and C, in an anti-parallel fashion, that is 5' to 3' in the other direction. So in a typical Watson and Crick GC pair or CG pair, you would have this, where this oxygen here is a hydrogen bonding to this hydrogen on the amino group. Here there's a hydrogen on a nitrogen that is hydrogen bonding to this nitrogen on the cytosine. And then down here, this oxygen, carbonyl oxygen, is hydrogen bonding to this hydrogen on this amino group in the guanine. In a typical AT pair, although it could be an AU pair also in RNA, you have up here this carbonyl oxygen, hydrogen bonding to this hydrogen on the nitrogen group over here, and you have this hydrogen on the thymine or uracil on this nitrogen group, hydrogen bonding with this nitrogen in the adenine. And if you flip back and forth between these, you can see how the hydrogen bonds change and how these can pack one on top of the other. This diagram shows those hydrogen bond lengths, and so you can see all of them are between about 0.29 and 0.3 nanometers in length, so they're fairly uniform in their distances. The reason you don't have a third one for an AT pair, which is fortuitous actually in the case of evolution, is that this carbonyl oxygen has nothing to pair to on the adenine ring. Here's a longer length of a double-stranded DNA where you have the 5' end, which usually has a phosphate group on it, and then the 3' end would be down here with a hydroxyl end. And over here you would have a 5' phosphate on one end and then a 3' hydroxyl on the other end. Because these phosphate groups are negatively charged, then this outside portion is hydrophilic, and that is then in contact with the aqueous or water part of the nucleoplasm or the cytoplasm in those cells. And the central part with these bases is hydrophobic, so those are tightly compacted on the interior of the molecule. DNA can take on several forms. The usual what's called the B form DNA or bDNA looks like this, and this is just showing three different types of models for that. But you can see this double helix form where it's a right-handed helix. So if you take your right hand and curl your fingers around towards your palm, then your thumb is pointing upward, and that's called a right-handed helix. And you can see there's a major groove here and then a minor groove here. The major groove is where many of the proteins will attach to the DNA. You can also see that the bases are compacted on the interior of the molecule and that those phosphate groups are sticking outward, and those would be then attached usually or in contact with water molecules and other hydrophilic molecules. Also, any positively charged ions would also be attached to DNA in those positions. So sodium is often found, potassium is often found attached to DNA as well as other positively charged ions. If you change the conditions to higher salt concentrations, the DNA will kind of squeeze together even further, hiding those bases from those positive ions, the salts. And so what you get is a more compact DNA with a little tiny hole in the middle but you very much compact that. So the DNA actually becomes shorter in length, in contour length, but a bit larger in its diameter. Under some conditions, especially when you have high levels of 5-methylcytosine in the DNA will flip around and become a left-handed helix, and that's called Z-form DNA. That's at the end over here. So that would be as if you took your left hand, curled your fingers around towards your palm, and then your thumb would be pointing in that direction. And so that's a left-handed helix. And now we have the bases really exposed on one side, and then the phosphate groups and the backbone on the other side of the helix. The most common place that you would find this would be in regions that are not being transcribed. So these would be regions that are not actively being expressed or transcribed. Here's another set of models of DNA, both B form, A form, and Z form, just to accentuate, to show you where some of those different pieces of the molecule are. So in typical B form DNA, then you have the phosphate groups on the ribose and the backbone around the perimeter of that molecule, and the bases are inside. In A form, you get a little bit more compaction of the molecule, so that then you have a narrower band of the phosphate groups, and then the bases are on the inside, but there's a narrow hole going down the center of that molecule. And then you have Z-form DNA here, where essentially on one half of the molecule the bases are exposed, and on the other half of the molecule, the other side of the molecule, you have the sugar phosphate backbone. And so the B form is the normal form that you find in organisms, especially in the DNA when it's being expressed. It will primarily be in this form, and that's mainly because the proteins, the DNA binding proteins and DNA and RNA polymerase recognize that form of DNA, and they don't recognize the other forms of DNA. Although on the surface you might think that RNA is very similar to DNA, it's not. It has some similarities and some major differences. So RNA tends to be mainly single-stranded, although it folds up in different ways. It can form double helices for short distances, usually 7 nucleotides or shorter. But overall it has a greater range of conformations than does DNA. So you can see this up here. This is the GNRA loop. So this would be a G and then anything R, meaning an A or a G, and then an A at the end. And often these are just G, A, and these are functional. These have functional properties in that they attach hydrogen bond to other regions of the molecule. The same RNA or a different RNA. Over here, here's somewhat of a model of what this small section of RNA looks like, where you have now a UG pair rather than a UA pair. And this solid circle in the middle means that that's a standard Watson-Crick pairing. Down here there's another GU pair, and that's a standard Watson-Crick pairing. But then you see these other symbols in here, and those stand for pairing of different types, which I'll show you in a few minutes. But not only can you get base pairing along the Watson-Crick edge, as in DNA, but you can get pairing between the sugar edge and the Hoogstein edge, which is the other side of the base, the adenine AGC or U, and they can pair in different ways. And then you see this particular molecule, you've got some crossing over of that molecule due to specific interactions between the bases, which are not the Watson-Crick pairs, but they're longer range interactions as well. still based on hydrogen bonding, but bonding to different portions of those bases. So RNA can take on many shapes, and there are non-standard connections between the bases as well, including this one, which is the cap on mRNA molecules in eukaryotes. And then this guanosine, which is on the end, it's a 7-methylguanosine. So on the 7 nitrogen group, the 7 position on the double ring, you have a methyl group, and then instead of being connected in a 5' to 3' sense with the rest of the mRNA, it's connected through 3 phosphate groups in a 5' to 5' way. So this is only recognized by a few different proteins, including what's called the cap binding protein, which recognizes this, which takes that mRNA then through the nuclear membrane and attaches to the small subunit of the ribosome. And then often, but not always, this first nucleotide on the mRNA is an A, and the next one is a G. And then RNAs can bind to other RNAs in different ways. So as I said, there's this Watson-Crick edge, which is present in all of those, and that's the edge that bonds DNAs together, DNA nucleotides together. In RNA, you have 3 possible edges: the Watson-Crick edge, a Hoogsteen edge up here on another part of that base where you have possible base pairing, and then the sugar edge down here where you have possible base pairing with other nucleotides. So instead of essentially one geometric family in DNA, DNAs are joined together in a standard cis Watson-Crick manner that is cis with regard to what side of the molecule the sugar group is attaching to the base. In RNA, there are actually 12 geometric families, and these 6 shown here, and then either cis or trans in each. So you can have the Watson-Crick edge on one, hydrogen bonding to the Watson-Crick on the other, Watson and Crick binding to Hoogsteen, Watson and Crick binding to sugar, Hoogsteen to Hoogsteen, Hoogsteen to sugar, sugar to sugar, and again, cis and trans for each one, so that then you have 12 possible geometric families of joining these together. Therefore, there are several AU pairs, several types of GU pairs, there are AC pairs, there are A pairs, there are GG pairs, etc. So many different possibilities for RNA for joining the nucleotides together. And because you do have several edges with which to hydrogen bond to, base triples are possible, and they have been found in RNA. So here's a UAU base triple, and these little stars are where the sugar bases are attached. So there's a standard AU pair, and then there's another base joining in here, another uracil, which is interacting along its Watson-Crick edge to the Hoogsteen edge of the adenosine. Here is a CGG base triple. CGC base triples also are found commonly. And again, this is a typical CG base pair here, as well as another guanosine joining in here along its Watson-Crick edge to the Hoogsteen edge of the guanosine. So RNA does many different things. So RNA molecules do many different things. As I've already shown you, ATP, which is an RNA-based molecule that transfers bond energy around the cells. Another one is NAD+, nicotinamide adenine dinucleotide. So there are 2 nucleotides here joined together in a 5' to 5' direction. Here's the adenine here, and this is nicotinamide down here. This is important in redox reactions. That is reactions where you're moving around electrons and protons. otherwise known as reduction, oxidation, or redox reaction. So very, very important, and these molecules have been around for billions of years, probably 4 billion years or more. An intracellular messenger here is cyclic AMP, again based on RNA. It's the ribose sugar is cyclized with the phosphate, and then there's the AMP. This interacts with many different proteins and is considered an intracellular messenger in that it signals many different proteins to become active usually. Also, there are many more bases in RNA than you find in DNA. So DNA essentially has 6, right? A, G, C, and T, as well as methyl A and methyl C. But RNA has many more than that, and I've already shown you the 7-methylguanosine. That's the cap on mRNA in eukaryotes. But there's also N6-isopentenyl adenosine. So it has a pentenyl group here hanging off the adenosine. Here's N6-methyladenosine, which I talked a bit about, quiosine, and then yosines on this R1 and R2. You have a variety of different moieties hanging off of those. And here are some more. Here's inosine, which interestingly can pair with anything. It can pair with A, G, C, or U. Xanthosine, 7-methylguanosine, which I talked about as being the cap on the mRNAs. Here's pseudouridine. This is 4-thiouridine, having a sulfur instead of an oxygen in that 4 position. Dihydrouridine. This is one that's found in many different types of tRNAs. And then 5-methylcytidine. So unlike DNA, RNA is much more complex, so that approximately 120 different bases have been described in RNA, while only 6 are known for DNA, that is A, G, C, T, and then methylated methyl A and methyl C. So while DNA primarily carries the genetic information, RNAs can do many things, including carry genetic information in some viruses. So some RNAs are ribozymes, that is, they're enzymatic RNAs. They can fold in dynamic ways, and they're actually capable of changing their form in many cases. Some can transfer energy within cells, such as ATP. Some participate in redox reactions, such as NAD, NADH, NADP, NADPH. Those all can transfer electrons and protons sometimes. Some synthesize polypeptides. Those are the ribosomes, which are the large subunit of the ribosome is also a ribozyme. Some deliver amino acids to the ribosomes. Those are tRNAs, transfer RNAs. Some transfer information to synthesize proteins. Those are the mRNAs, or messenger RNAs. And then some control gene expression, and we'll talk about those small RNAs that can control expression and longevity of mRNAs. So they can control expression. And as I stated in an earlier video, RNAs have been around longer than DNAs, and they performed many, many functions in the initial cells that were on Earth probably 4.2 billion years ago, starting from 4.2 billion years ago. But they continue today. So many of the functions in your cells still rely to a great extent on RNAs. Part of their versatility goes back to this, in that they don't form just standard Watson-Crick base pairs, but they can form many, many different interactions. And the many different types of nucleotides, the 120 different ones, can affect how the RNA molecules fold together, how they interact with other molecules, including other RNAs and proteins. and their ultimate structural conformation. So here's a little more detail in how RNAs are able to pair with one another, one nucleotide to another. Here's adenosine here with the Watson-Crick edge, Hoogsteen edge, sugar edge. There's guanosine, the same with its edges. And then the pyrimidines over here, uracil and cytosine, with the Watson-Crick, Hoogsteen edge, and sugar edge. So here on top is a standard cis-Watson-Crick AU pair, again with the sugars on the same side of the molecule. And here's a trans Watson-Crick, Watson-Crick, with the sugar groups sort of pointed away from each other on opposite sides of that molecule. And here are the 12 different families. The cis ones have the filled symbols down here, and the trans have the open symbols down here. So here's a cis Watson-Crick, Watson-Crick, and that's a standard Watson-Crick base pair that you'd find also in DNA. But then a trans Watson-Crick, Watson-Crick would be this way, where the sugar groups are on opposite sides of that molecule. Then here is a Watson-Crick Hoogsteen cis conformation here. And you can see these filled symbols, the circle standing for a Watson-Crick edge, and the box standing for a Hoogsteen edge. And then here's the trans version of that. And then up here is a Watson-Crick sugar edge cis conformation with the triangle then standing for the sugar edge. And down here is the trans version of that. And then here's a Hoogsteen, both cis and trans, a Hoogsteen sugar, both cis and trans, and a sugar, both cis and trans. So that adds up to 12 geometric base pair families, and there's actually 18 pairing relationships, because with the ones that have different edges, that is Watson-Hoogsteen, Watson-sugar, Hoogsteen sugar, you can have a couple of different orientations based on which one has the sugar edge and which one is using the Hoogsteen edge, etc. So there are 18 pairing relationships. This leads actually to, if you have a molecule that's 60 or 100 bases long, leads to a lot of different possibilities as to how that folds. And this is a table just indicating some of those symbols, the interacting edges, whether they're cis and trans, and whether that leads to local directionality of the other strand. So if these things are paired, then you can have the standard anti-parallel orientation of the 5' to 3' direction on one strand and 5' to 3' direction in the other strand, whereas some of these other ones can lead to parallel strands. So you can have a 5' to 3' in one direction, and on the other strand it can also be 5' to 3' in the same direction. So again, many different possibilities exist for RNA molecules to fold. So in DNA, it's normally a double-stranded anti-parallel molecule with the standard cis Watson-Crick edges meeting to form those hydrogen bonds. In RNA, many different structures are possible. So you can have single-stranded regions, which are quite common. You can have short double-stranded regions, which are also common. You can have a bulge loop consisting of one or more nucleotides, or an interior loop that looks like this, or a hairpin with a junction joining that to the other part of the molecule. These are all common in transfer RNAs, tRNAs. This is the secondary structure. So here's the acceptor arm up here with the amino acid attachment site, which is a CCA nucleotide at the end. This is the D loop, mainly standing for dihydrouridine, because often that loop has a dihydrouridine within it, one of those different bases. Here is the anticodon loop down here that contains the anticodon that attaches to the codon in the mRNA. A variable loop, and it is quite variable among the tRNAs. And then a T loop, named primarily because it has a thymine within it rather than a uracil. It does have uracils also, but it has a thymine within it. Here's the 3D structure of that. So you can see not only do you have some short double-stranded regions, but you also have regions that interact with each other long range. So the T loop has some interactions with the D loop. So there are some hydrogen bonds, essentially long range, but they're actually, when you fold this up, short range. And then the D stem here, and then another short loop here of the double-stranded loop here, and then the anticodon exposed down here. So this shows a few more of those long-range interactions. So there's interactions between the D loop and the T loop, and with the T loop and this a short double-stranded region down here, as well as this loop, part of the T loop here, interacting with part of the double-stranded region of the acceptor arm. And then you have some interactions here in the anticodon loop between some of the bases in that loop. And then the complex process of translation of the mRNA into a protein, almost all of those functions are performed by RNA. So up here you have the messenger RNA that carries that message as triplet codons. There are initiation factors and elongation factors that bind to that. And then you have the ribosomal RNAs, which become the small subunit ribosomal RNAs, large subunit ribosomal RNAs, as well as some 5S RNAs that come from elsewhere to build the ribosome. And the large subunit here is the catalytic part of the ribosome, and the small subunit really delivers the mRNA to the large subunit and also ratchets the message through the ribosome at a rate of 3 nucleotides at a time, which is the size of a triplet, of the triplet codons. And in order to bring those amino acids in, then you need the tRNAs, which are built. A CCA is added to that, to the 3' end. The amino acid is added to that using some proteins, which probably used to be RNA molecules, but are now, those functions are done by proteins. So each one of those amino acids is then brought one by one to the ribosome to be added together by peptide bonds to form either the peptides, the polypeptides called either peptides or proteins, depending on their size or length. And remember, RNAs at the same time are being used. You need GTP for this, some of these functions. You need ATP. You also need NADH, things like that. So other RNAs also are used in this process, as well as many or most cellular processes. So as I said, these are brought together, joining them as peptide bonds. So you have the L-amino acids are the ones used in biological systems, and this was probably an early selection, back some 4 billion years, to use only the L-amino acids and not the D-amino acids, although there's a minor component of D-amino acids in some, and there are a few that don't have stereoisomers. So, but 99.999% of the amino acids in proteins are L-amino acids. And these are brought together by the ribosome in peptide bonds, so water is given off in the reaction. And these are added by using the genetic code, which is set by the aminoacyl tRNA synthetases, in that they are attaching the amino acids to specific tRNAs that have a specific anticodon that then is interpreted by the ribosome by comparing with the mRNA, right? So we'll talk in much greater detail about this part in a few videos from now. But just to foreshadow that a bit, these amino acids on the bottom row, as I've already talked about in a previous video, are among the smallest and simplest amino acids, and probably were the first amino acids that were used in biological systems. And then you go up on this side, and these are mainly the 10, 1, 4 down here, 6, 8, 10. These are the 10 amino acids that were available on early Earth. So the genetic code probably started down here, moved up in this direction, and then some of the last amino acids to add to the genetic code and proteins were up here, tyrosine, tryptophan, and cysteine. And the color coding on here, the orange ones up here are the nonpolar amino acids. The purple ones are the polar ones. The blue color are for the basic positively charged amino acids, and the green ones down here are for the acidic or negatively charged amino acids. So you can see there is some structure to this genetic code, and again, we'll talk about this in much greater detail in a few videos from now. So from translation, of course, you get proteins, and proteins have between 50 amino acids and hundreds of amino acids in length, depending on the protein. And they perform many of the functions that RNAs used to perform, and many more. So once translation was invented or evolved in these organisms, then it really took off, because you could have much greater variation and diversity in the functions, and proteins are usually more stable than RNA, so it provided a couple of different advantages over organisms that were purely RNA based. So the categories of proteins, of course, are these. They can be structural, such as microtubules, enzymatic, such as lysozymes, signaling, such as insulin, transport, like hemoglobin transports oxygen, storage, such as ovalbumin, storing essentially amino acids, regulators, such as the lac repressor and transcription factors and things like that, kinetic, such as actin and myosin, and flagellants, receptors such as rhodopsin, special purpose proteins such as antifreeze proteins, and recognition proteins such as immunoglobulins. They vary quite a bit in size, so here's some protein diversity. Here's the large ribosome up here, which is RNA, not protein, and there's the tRNA in relation to that. Here's an antibody, an immunoglobulin, a fairly large protein. Actin filament, also a fairly large protein. Here's myoglobin, which is an ancestor really of hemoglobin, which consists of 4 of those chains, so you can see the increase in size there. These larger ones are viruses, but then out from the virus you can see the individual proteins that make up the protein coat. So this gives you an idea of the relative sizes of some proteins compared to these multimolecular structures that we call viruses, these infectious particles. So as I stated before, in this class I won't discuss proteins much more than this. I will discuss some specific proteins, but in another class that I teach in cell biology, I go into a little more detail about some of the protein diversity. But in this class I will stick primarily with DNA and RNA. Although proteins are nonetheless important, it's just that there's just too much to talk about when we talk about DNA, RNA, and then try and add proteins to it. Although I will say that, so early on, cells were based primarily on RNA and some other molecules. And then shortly thereafter, they incorporated peptides, which are small proteins, but didn't use ribosomes at that point. Then ribosomes evolved to produce proteins with more fidelity and at a higher level. Once that happened, then a lot of the functions that originally were carried out by RNAs were taken over by proteins, again, because the proteins were more stable and they could be produced in higher numbers, in higher fidelity than they could just by using RNA molecules. So another important set of molecules in living systems are membranes, and they self-assemble. And also, many of these can be produced, or probably were produced early on Earth by abiotic means without using biology. So they could self-assemble and essentially encapsulate things so that there was an inside and an outside, which is absolutely necessary for life. Most are phospholipids, so they have a fatty acid region, which is hydrophobic, usually two fatty acid tails. In the case of archaea, they're isoprene units rather than fatty acids, which are mainly saturated and unsaturated. That is, they have a double bond in at least one portion of these chains. So because these are hydrophobic, then they associate with one another, and they exclude most molecules so that you can have an inside and outside of a cell. And not many things can penetrate through that hydrophobic layer. Those are connected through a glycerol molecule, which is a 3-carbon molecule. So two of those are attached through ester bonds, and then the third carbon is attached usually to a phosphate group and some other molecule as well at the top, which often is a molecule called choline. So in that case, it would be called phosphatidylcholine. And that end of the phospholipid is polar, that is, it will dissolve in water. It is hydrophilic, so that then these associate with their tails pointing towards one another, their hydrophobic tails, and their hydrophilic heads either pointing to the inside aqueous part of a cell or the outside aqueous portion of the environment. And then one other important set of molecules are carbohydrates, including sugars and polysaccharides. So there are monosaccharides such as fructose and glucose, which are used quite a bit in biological systems. And then there are disaccharides such as sucrose and lactose and other disaccharides, which can be broken down into those monosaccharides again. And then there are polysaccharides. Some of these are storage type sugars, and then others constitute cell walls and things like that. So in plants, you have amylose, which is just chains of glucose molecules. In animals, such as us, we have glycogen, much of which is stored in our liver. And that's not only consists of chains of glucose, but also side chains of polymers of glucose as well. So this one, in this case, you have just chains and chains and chains of polymers of the monosaccharides together. In plant cell walls, they have cellulose, and again that's made up of individual repeating units, polymers of the monomers of the sugar to make up cellulose. So that's by no means an exhaustive list, but includes some of the very important molecules that were available even early on Earth to create some of the first organisms, the first life on Earth, and then lead to the diversity of life on Earth. And again, I'll concentrate primarily on the nucleic acids, but I'll mention some of the other molecules from time to time. Okay, this is the end of the video called Molecules of Life, where I spent most time on DNA and RNA, spent a little time on proteins and some of the other molecules. And I'll go into some more detail about specific RNAs and then specifics on DNA in subsequent videos. So thanks for listening and watching, and I will talk to you later.